because we don't care about that. I want to introduce you to Dr. James Bieker. He comes to us from the Mount Sinai School of Medicine, where he's a professor there. And he got his bachelor's degree at St. Louis University in chemistry, and went on to Northwestern University, where he got his Ph.D. in biochemistry. And he did postdoctoral fellowship at Wash U, and also the Rockefeller, it looks like. Is that correct?
Yeah.
Okay, perfect. And I don't know whose lab that was in. I should have asked that part.
Bob Rader.
Okay, so are you from the St. Louis sort of area?
Yeah, that's where I was born. Yeah.
Okay, cool, awesome. All right. Well, we want to give the time to you for this presentation, and I want to ask people to please mute themselves if they're not asking a question. You're welcome to keep your screen on to see things. Dr. Bieker has agreed to questions during the middle, and you can populate them in the chat, or you can raise your hand, or you can, if there's a pause in the talk, you can interrupt and ask a question if you have one during the presentation. So thank you so much again for your presentation today.
Oh, sure. Thank you very much. I'll go ahead and share my screen here. Yeah, so this is great, and it's just nice to see. I'm seeing here in the video a number of familiar faces here. That's great. So so what I'm going to focus on today are some of our studies related to the erythroblastic island, and particularly transcription factor that we've been working with for many years, named EKLF. So I'll give you a little bit of background on this particular protein and where it came from. Now we've been interested for a number of years at this process of can you see my arrow, my pointer? Okay, well, let me know if you can't see where I'm pointing.
Sorry, I was muted. Yes, we can see it.
Oh, okay,
clear.
Great.
It's clear.
Thank you. And so we've been interested. This is sort of, I know it's slightly out of date because it's a little more complicated, but at least makes the point about stem cells, which are able to repopulate themselves, but then also bifurcate into a number of other lineages, eventually giving rise to the blood cell lineages. And so what we were interested in is in this process over here of erythropoiesis. What makes this unique compared to other lineage trajectories here? And what we did a number of years ago is we compared the RNA sequence information from erythrocyte cells using the murine erythroleukemia cell line as our model. And from that, we subtracted genes present in the macrophage cell line called J774. Now, this was quite a few years ago. This was in the pre-array days, the pre-seq days, and this was done by actually making cDNA and then subtracting, doing an RNA-DNA hybridization and using hydroxyapatite to separate these molecules, and eventually cloning what was left over into these old lambda vectors that then we eventually went through. And so we were very interested in doing that process. We identified a number of genes unique to this erythroid lineage. One that grabbed us in particular after we finished the sequence analysis was a protein which we eventually called erythroid Krüppel-like factor because of its homology of its zinc fingers to the Drosophila Krüppel protein. Now, this ended up being very convenient because this fell into the C2H2 cysteine, histidine, zinc finger family. And at that period of time, there was a lot of interest in the structural biology angle, that is using the X-ray and the NMR data to say, can we take the structure of a finger and predict what it binds to? And what had been discovered was that each of these C2H2 zinc fingers interacts with a triplet, and we came up with a triplet predicted DNA binding site, which this is the C-rich strand, but is primarily on the G-rich strand, which is where the interaction is. And so okay, and so what was fortunate for us is that this matched an already known important promoter element of the adult beta globin gene, the so-called CAC element. This was fortunate because this put us in touch with a real target gene. We didn't have to rely on artificial targets to get us through. And one of the other advantages of this is that in the thalassemia field, there's a lot of beta thalassemia causing point mutations throughout the gene, but also upstream of the gene. And we knew of 3 at the time, which we tested individually, and we found that if we had these individual single point mutations, we found that in our transactivation assays, which was done by co-transfection into a reporter cell line, even though our wild type could transactivate over 10-fold, all of these had lost transactivation activity. And when we looked in vitro to look at DNA binding, it turned out that each of these point mutations decreased binding by 40 to 100-fold. So this told us that we isolated an interesting factor that bound to an important promoter element that likely bound to this. Now, to try to get an idea of whether it could be involved in globin gene regulation, we did this experiment in collaboration with Tim Town's lab. Now, I should remind you that the globin genes are under developmental regulation, and indeed you have the fetal globin gene, which is linked in cis to the adult beta globin gene. And as I showed you, it interacts very precisely with the adult beta globin gene, but we already knew that the CAC element wasn't exactly right in the fetal globin gene, that is the gamma globin gene. So what we did is we did a co-transfection assay where we had the gamma globin gene upstream of one promoter, the luciferase promoter, linked in cis to the beta globin gene upstream of the CAT promoter. We co-transfected this with or without a EKLF expression vector. And what we found was that the adult beta globin gene was knocked off the roof, over 1,000-fold activation, whereas the gamma luciferase reporter was really much less. This suggested that this may be an important switching factor involved in human globin gene regulation. I should point out that we were also lucky that the K562 cells were available. This is an erythroleukemic cell line from humans that, fortunately for us, expressed very little to no EKLF. So as a result, it provided a very nice erythroid type background, and yet a cell which was not expressing much endogenous EKLF, so we could actually look for these types of effects. So this provided then the basis for a lot of the subsequent experiments that I'll be talking to you about. Clearly, then I'm gonna then summarize just some additional studies that really puts this all in context. One is the prediction was that it plays an important role in globin genes. Well, we wanted to show that. And we were doing these experiments with Frank Costantini to do the knockout in the mouse. Unfortunately, we were scooped by 2 groups, by the Orkin group and by the Frank Grosfeld group, which was too bad for me, but scientifically it was great because, as you can see here, I think you can tell right away which one's the littermate that's a knockout versus the het. And what was interesting was that when you look at the adult globin gene expression, you find that there's a lot of problems here. There's a lot of dysregulation of the adults. And indeed, when you look at the yolk sac genes, they look pretty normal. Now, we know in subsequent experiments they're actually not expressing well, but at the phenotypic level, they look pretty good. And indeed, this turned out to be a very critical gene because this was an embryonic lethal mutation. They died by day 15 of a profound beta thalassemia. So this indicated we had the molecular data on what it interacts with, but this also indicated then that it also plays biologically an important role, and we didn't run into redundancy issues, at least at the adult stage. So some other characteristics that I do want to describe, because this will be important in subsequent studies, it exhibits an extraordinary level of tissue specificity. So this was the original northern blot when we first identified the gene. And this DS19 is the MEL line, and J774 is the macrophage line. It's a very clean difference in expression. And when we did an organ blot from adult mouse tissues, we found that it was only expressed in bone marrow and spleen, which are exactly the erythropoietic lineages in the adult. So this suggested a very strong tissue specificity. When we look at early development, it only comes on in the early head fold stage of the developing mouse embryo, in the blood islands of the yolk sac. It then switches expression to the fetal liver. And then in the adult, it's only expressed in the red pulp of the spleen. So again, retaining this high level of restrictedness in its expression pattern. When you look during hematopoiesis, it's not really expressed in the long-term progenitor, but it becomes expressed in the multipotent progenitor. Remains low in the common myeloid progenitor, but then becomes very high in the MEP, the megakaryocyte erythroid progenitor. Now here, interestingly, it really diverges such that it becomes very low in the megakaryocyte progenitor, but remains very high in the erythroid progenitor, such that by our analyses, there's about an 80-fold difference in expression between these two progenitors. And so as a result, it plays a critical role in the red cell, but it does not play a role in the megakaryocytes. Yet it is present in the progenitor to these two types. And one question that we've had for a number of years that we would like to follow up on at some point is what accounts for this bifurcation, this bipotential decision making that's going on here. So, since my background was much more in biochemistry, one of the things we were very interested in doing is how does it do what it does? What does it interact with? And this summarizes actually a lot of work. And Yvette and I wrote a review that covered a lot of these topics. But essentially, what we found is that it interacts not just with transcriptional proteins such as the TF2H subunit and TAF9, which interacts with TBP, and that accounts for its transcriptional activation. But what we found is it interacts with histone modification proteins, such as P300 and CBP, and also with the SWI/SNF chromatin remodeling proteins. So we feel that EKLF plays a role in integrating all of these diverse signals into giving rise not just to active transcription, but to an open chromatin domain. It also does this by interacting with a histone H3.3, with a chaperone for that. And other experiments showed that it itself, EKLF itself, gets modified by some of these interactions. It gets phosphorylated, which was not a surprise, but it also got acetylated. At that point, it was only the second transcription factor found to be acetylated by virtue of its interaction with these HAT proteins. It gets sumoylated, and there's a lot of other interactions that are going on here by virtue of the fact that it gets modified. This then, not surprisingly, then Frank Grossfeld's group showed that it plays an important role in the three-dimensional structure. So this so-called active chromatin hub requires EKLF for the proper interaction of this upstream hypersensitive site, which is also down here called the locus control region, with recruiting the beta globin gene. Whereas if you don't have it, you just don't have the adult beta globin genes expressed. And finally, with Peter Fraser, we showed that in the nucleus, it also plays an important role in forming these transcription factories, which are not just KLF1 here, these roughly 40 transcription factories, but also recruitment of RNA polymerase II. And also within these factories, Peter showed that a lot of these target genes, such as the beta globin gene and other EKLF targets, are also localized here. One thing I'm not going to discuss because we actually still not sure what's going on. You see a lot of red signal in the cytoplasm. There's a lot of EKLF in the cytoplasm, and we've discovered this, I think, 15 years ago, and it's still unclear what is it doing there. But anyway, that's a topic for other discussion. So one of the exciting things that's popped up in more recent years is that indeed there have been some human mutations that have been mapped to EKLF, which have really enabled us to focus on this whole set of, some of these are benign, some of these lead to haploinsufficiency, and one of these leads to congenital dyserythropoietic anemia. But the bottom line is this really firmed up the idea that KLF1, as it's also called, plays a central role in basically everything related to hematopoiesis, to erythropoiesis. Not just globin gene regulation, which was I've been really telling you about, but also cell cycle regulation, a lot of the iron and heme related targets that show up there, and also the blood group antigens are also altered in their expression. So, excuse me, this you know, it's sort of, this is sort of, I call this the Aristotelian view of KLF1 activity. It's the center of the red blood cell universe. But one of the other things that has come up from this in terms of globin gene regulation is that EKLF is upstream of what a known repressor that some of you may have heard of because it's been analyzed, and BCL11A, a repressor of gamma globin expression, is being attempted to be modified to then see can you remove the repressor and therefore reactivate gamma, because that would be fantastic for hemoglobinopathies such as beta thalassemia and sickle. EKLF is upstream of BCL11A, and indeed it also regulates other repressors, such that this leads to whether KLF1 can be engineered as an aid for hemoglobinopathies. This is something we're actually working on. The complication, the good thing is that it's tissue restricted, so one could envision this. The bad thing is that we can't knock it out because we'll totally screw up the red cell. So this would require a very fine regulation of its levels, and this is something we're interested in, but it still has a little ways to go. But so essentially, to summarize this introduction, I've given you sort of quite a bit of background of a number of years of work on EKLF, its zinc fingers, the fact that it interacts with an important promoter element, that it's restricted in its expression, it interacts biochemically with a lot of factors, and then also its biological disruption leads to various aspects related to red blood cell and globin gene regulation. But a number of years ago, we were interested in using embryonic bodies to try to also identify other aspects of the early role of EKLF in development. And embryonic bodies can be formed from embryonic stem cells and methylcellulose, and then one can analyze a variety of these embryonic bodies for various lineages, and I think most of you know, it's just because this is also done with humans, human embryonic stem cells, to really look at in the dish what type of aspects might be interested in. So the aspect we really went on, started these experiments quite a few years ago when Gordon Keller was still at Mount Sinai. And so this was basically take these developing embryonic bodies, dissociate them, and then look for hematopoietic progenitors, precursors. So we did these types of experiments, and this was when Deepa Manwani was in the lab. Embryonic stem cells, differentiate them, and then disaggregate them. 6 days later, look at these colonies in methylcellulose. We were very surprised to see the colonies look like this. Instead of strictly a red cell colony, what we had was a central macrophage surrounded by these erythroid cells at various stages of maturation. So we thought this was unusual. So we also then went back and did this a little more extensively, where we took definitive embryonic bodies, did lineage depletion, did a FACS sort, and sorted for CMPs and MEPs, put these in methylcellulose, and got these very nice red BFUE-like colonies. And again, when we picked them and cytospun them, we again found these central macrophage involved in all of these things. Now, the reason this was striking is because this looked like a classic erythroblastic island. These are pictures from, from 30, 40 years ago, and very much similar to what we were looking at here. This electron micrograph is shown on the right. So what's the big deal about erythroblastic island, and why is this something that we wanted to look at? These were discovered in the late 1950s by Marcel Bessis in France, and followed up with a number of studies with Mohandas Narla, who some of you may know, when he was then, after he'd gotten his engineering degree, I believe, went to do a biological postdoc in France. And it turns out that if you look in vivo, maturation of these blood cells occur within these specialized niches called erythroblastic islands, central macrophage surrounded by erythroid cells, exactly what I've been showing you. Now, what is the thought as to what these cell surface interactions do? Well, it's thought that the macrophage provide a positive stimulatory growth, cytokines, and also anti-apoptotic regulatory effects on the developing erythroblasts. It's thought that they supply an important level of iron for hemoglobin synthesis, and also the phagocytic properties of macrophage leads to a DNase-dependent breakdown of the extruded condensed nuclei, and therefore it promotes enucleation. So what you end up having is you have this central macrophage, and you have erythroid cells of various maturation levels. But then what gets released is thought to be a young reticulocyte. That is a cell that's already had its nucleus removed. So given what I've told you about EKLF and about the way we sort of got into this, we thought, well, let's take a look at what these cells look like and whether knocking out EKLF has anything to do with it. Again, these are the erythroblastic islands, and when we look at F480, using F480 as the macrophage marker, you can see that the central macrophage is surrounded by a lot of nucleated cells, and some nuclei are inside the macrophage. When we did this isolating erythroblastic islands from EKLF knockout cells, we saw a very different picture where we saw these engorged, basically macrophage cells. As you can see here, the nucleus is really sort of within it. Very different aspect to the macrophage function. This looked very reminiscent of the DNase 2 knockout in macrophage, where the macrophage can't nuclease the DNA and therefore don't release things properly. But this led to the question, well, is EKLF expressed in these F480 macrophages? So what I'm going to show you are a number of approaches that we used to try to really nail this down. One was, by this point in time, when Felix Lohmann was in the lab, we had been doing a lot of analyses to look at how EKLF itself is regulated, because as I told you, it's really tissue restricted. So how does that happen? And he had established an embryonic body, embryonic stem cells, where we had this inserted reporter where we had the promoter of EKLF, which is this region upstream here, which we already knew contained an erythroid-specific hypersensitive site and an enhancer, upstream of the GFP, knocked into the HPRT locus at the homing site. And when you look at embryonic body differentiation, you can see that the GFP green totally mimics the expression of the endogenous EKLF. So we felt this was a reasonable reporter to use for some of these experiments. So we established a mouse line containing this reporter from these embryonic stem cells. And when we look at these cells, we see one piece of evidence for EKLF present in the macrophage. And that is, if you look at these F480 positive, EKLF positive cells, that is GFP positive cells, about a third of them have EKLF expression. We then really firmed this up by directly doing immunofluorescence. This was Shui Li, who's in the lab, where we showed by looking at individual cells that there are overlap of cells that contain both red and white signals. Not all of them, consistent with the FACS data. You see some cells that are only red and some cells that are only white. But again, we do see individual cells that have overlap of F480 and EKLF. A third level was a much more recent study from Julie Ahn's lab at the New York Blood Center, where she was investigating the role of EPO receptor in macrophage in terms of the role, particularly in the adult mouse, but also in the context of erythroblastic islands. And what she found is that KLF1 was highly enriched in the EPO receptor GFP positive cells. That is, the GFP in their case was a reporter for the EPO receptor. You can see here that these cells were negative for TER119. So this presence of KLF1 was not due to contaminating erythroid cells in the macrophage. Finally, a totally different analysis done by Geisman's lab, identifying c-KIT, C45, these multi-faxed cells, and doing individual RNA-seq doing an RNA expression analysis during early development. What was neat here is that indeed these are fetal liver macrophage cells that are expressing, this is the RNA profile, this is the RNA gene here, EKLF gene there. You can see a few things. One is that it is expressed in these highly enriched cells, but what was really striking is that it was a dynamic onset and decline. I thought this was totally cool because this then was consistent with what I told you before, where if you take an adult macrophage cell, we don't pick up EKLF there. And that was initially one of the reasons we were thinking, well, what's going on here? But it turns out during development, we have a very different situation. So given these studies, and given some of the roles of EKLF in the red cell, because as I mentioned, it regulates iron regulation in the red cell. So one of the questions we tried to address here earlier was to try to see whether it could regulate iron in the macrophage. Now, unfortunately, I'm not going to be able to finish this part because it's I think there's still some issues that we need to resolve. But we were able to establish the floxed EKLF mouse. We then crossed this with a mouse that had been already crossed with an EKLF het knockout mouse. So this was a CSF1 receptor, which was under estrogen receptor control. So this was based on the fact that it's been widely used in macrophage studies. So when we did this, we then finally, after the variety of crosses, which I'm not going to go through, we have estrogen receptor responsive Cree line that then we were able to do a tamoxifen injection, wait 2 weeks, and then isolate the red cells and look at their properties, particularly the critical blood. I'm sorry, isolate the circulating blood and look for the mean cell volume of the red cell and the mean cell hematocrit hemoglobin levels. What you see is that there's a significant difference in the MCV and the MCH only in the blue, which is this particular line here. The two control lines didn't have this this type of effect. Now this is intriguing because low MCV and MCH are suggestive of a systemic iron deficiency in these mice. The reason I'm a little concerned about this, well, actually, what I should also say in support of this was that when we looked at the transferrin receptor, for example, levels in the knockout versus the wild type F480 macrophage cells, we do see that there's a significant drop in the transferrin receptor levels. But what I was concerned about was that this was really a very small, significant, but very slight. I want to point out that the y-axis doesn't start from 0 here, okay? So I'm trying to emphasize the difference here, but this is something I'll come back to towards the end, and I think there may be a better way to do this experiment. But be that as it may, this was something that then has piqued our interest and we're very interested in looking at. But to summarize this part, what we found was that EKLF plays a couple of roles in erythropoiesis. One, it plays a direct intrinsic role in erythropoiesis. It plays a role in the integrity of the red cells I showed you, but it also, there's this extrinsic role where it regulates macrophage gene expression. In particular, it may be VCAM1 and its ability to interact with the erythroid cell. But to really try to firm this down, we wanted to do RNA-seq of a number of these populations we generated. First of all, EKLF wild type versus knockout, and also of these GFP positive versus GFP negative cells, because the complement of these two would be, I think, a very powerful way to investigate this. But before we got to that point, we thought, well, let's take a look at the gene expression pattern just in the wild type, fetal liver F480 macrophage. There were a number of surprises here. One is that when we did a comparison with fetal liver erythroid cells, now these were enriched erythroid cells that had been cultured, and therefore these do not have any macrophage in them. That was one set. So this compares to the erythroid cell within similarly timed E13.5 fetal liver material. To look at, compared to macrophage, no one had done a macrophage analysis, but we then thought, well, let's look at the spleen F480 macrophage, because that we did have access to. And what we found is by looking at the way these RNA sequences are laid out, we find that these segregated separate from each other. And indeed, when we did a principal component analysis, we found that the erythroid cells and the spleen cells separated, which makes sense, but then also our fetal liver macrophage cells also separated from this. When we looked at macrophage markers and erythroid markers, we found that our fetal liver F480 cells were sort of in between, didn't fall into one category or the other. And finally, when we compared the RNA-seq directly from these expanding erythroid cells compared to our fetal liver and compared to the spleen, there were about 300 genes that were really differentially expressed, highly expressed specifically in this subpopulation. And
Oh,
some
we
of
have
these
a
included
question.
cell adhesion and cell movement molecules. We thought, okay, that's interesting. That adhesion stuff could make sense. So,
James, we have a question in the chat. Vanessa, do you
yes.
want to go ahead and ask it?
Oh my gosh, I didn't mean to interrupt the whole talk. I was going to ask
Oh,
at the
that's
end,
okay.
but
You're
if
okay.
I have the opportunity, sure. I just wanted to go back. Oh, sorry. I just wanted to go back to the slide where you're presenting the data with the macrophage specific to moxifin induced KLF1 deletion, where you
Yes.
saw the changes in the MHC and MCV pointing towards the iron
Yeah.
deficiency.
Sorry, I'm having problems going backwards here. Oh, here we go. Great. Yeah.
Yeah, and how you saw that reduction in the transferrin receptor 1, but I just wondered if you also checked the expression level of transferrin receptor 2, which I think sometimes can be considered sort of a readout for the iron level in the system.
Yeah. That one wasn't as dramatically affected as this one. But what do I mean by that? Certainly, I mean, here we're talking about like roughly a 7-fold drop. That was maybe at most a 2 or even less than a 2-fold drop.
Gotcha. Okay,
Yeah.
that's interesting. Cool. Thank you so much again. Sorry to interrupt.
No, that's fine. That's not a, no, I don't mind that at all. So this, the point of this analysis was then to tell us, okay, there is something a little different about the F480 macrophage from the developing fetal liver versus other cell types. And indeed, when we looked at the certain genes that are present here, we found that there was a certain number of signature genes that were present here. Now this is the comparison. I'm sorry, I didn't lead into this slide well. What I showed you before was the wild type. Now we wanted to see what is EKLF's role in this. And we found that there were a number of genes which were downregulated in the knockout of EKLF, which makes sense because typically we've looked at EKLF as an activator. So you might express that there's a larger number of genes that are downregulated when you knock it out versus than when you open it up. So this provides us with a signature set of genes that are downregulated when you remove EKLF. Okay. Now, I told you that we had established this EKLF GFP mice. So, so we thought this would be a good complement to what I just told you. So the genes I just told you, signature genes 1, are genes that may be directly downstream of EKLF. When we look at this is a perfectly normal mouse. It's just that we're monitoring cells that are expressing EKLF versus ones that are not. So we're not looking necessarily at the knockout effects. And when we looked at principal component analysis, we found that these were very nicely separated. And so we thought if we take this signature set gene 2 and overlap it with signature gene set 1, that is genes that are positive in the GFP case, but absent in the null case, we found that there were a number of genes, like 500 genes, that fell into this category and could be EKLF targets. This was further aided by doing what's called, I think it was called the Centrimo analysis, where you could look at these 500 genes and see if there's any enrichment for EKLF binding elements in the promoter. We did find indeed that there's an enrichment for KLF binding sites within these 500 genes. So you put this together and we thought, okay, we can actually use this to try to identify the subset of macrophage genes that are possibly involved in the island. So to do this, we sort of monitored the overlap of all of these expression patterns with the aim of trying to say, can we pull out a cell surface marker of, in addition to F480, which can then subdivide F480 cells into the subset that is involved in the macrophage in the island. And so from looking at the differences, as I've told you here, from the previous slide, focusing on genes that were up in the GFP positive and the difference in the range, we found that a potential enrichment marker was the adrenergic receptor type 2B, which was a surprise because that tends to be more in the neural cells. But when we had the RNA data from a number of different directions, and when we did immunofluorescence, we found that indeed the central macrophage nicely expresses ADRA2B in the cell. Thought, okay, great, but there are two things that we're worried about. One, as I called it, is the elephant in the room, which is this whole aspect of macrophage heterogeneity. I have to say, as the years have gone by, I've learned more about macrophage. I've been amazed compared to the erythroid cell, how heterogeneous we've got the situation here. And I think that's a problem. So
one more question in the chat.
Yeah.
Anna, do you want to go ahead?
Sure, sorry. I was just wondering at what stage was Cree-induced by 4-OHT at E85? Is that the model you?
I see. No, I'm sorry, that wasn't clear. No, that was done in the adult. So we just injected the adult and then waited 2 weeks. So it wasn't done embryologically.
Oh, okay. All right. Thank you so much.
Yeah. So now I think I'm talking slower than usual. I'll try to speed up a little bit. But basically, one of the things I do want to address here is this whole purification challenge, which relates to worries about cross-contamination. And this is something we've been very concerned about. And so in our experiments, we have taken care, and indeed we've talked about this in some of our publications, to morphologically verify that indeed we're looking at stringently sorted cells that morphologically look like they have one nucleus, that we can look at, use image stream to individually look at marker expression with each individual cell to make sure we've got clear fact segregation. And I think a very important thing is this use of this peptide marker, which we started using quite a few years ago based on Joel Chase's observations, that it blocks ICAM4 alpha-4 integrin interactions between erythroid and macrophage cells. So for example, here again, you see this, and this was, we talked about this a number of years ago, where you see this interaction. And if you include this FWV peptide, you decrease the colony formation. Indeed, you really decrease the interactions between erythroblasts and macrophage cells. So we routinely include this whenever we want to analyze some of these things. And now this was important because the next thing we wanted to do was single cell sequencing. So with the single cell sequence analysis, we tried to focus on making sure we avoided cross-contamination by virtue of all of these ways. When we did single cell sequencing, we found, again, not surprisingly, if you're anticipating heterogeneity, we could find 13 different clusters in the macrophage cells. These were distinguished very nicely based on heat maps at the RNA level. And indeed, when you look at the F480 distribution, all the clusters are expressing F480, but they are at different levels. Joe, you have a question?
Yeah. Go ahead, Joe. Joe Prakal. What's your burning question, Joe?
Right, no, can you hear me now, Jim?
Yeah.
Yeah, okay. So Doug Hicks described 8 families, I think in 2014, with compound heterozygosity. I believe these were missense mutations of EKLF1, who not only had a severe anemia and increased expression of embryonic globin, but they also had a pyruvate kinase deficiency. And there are 2 genes for pyruvate kinase deficiency. This was the gene which is erythroid specific, called PKLR. And I've never seen any other papers from that. And it really, do you have any insight on that there was dysregulation of globin expression, but also a specific defect of pyruvate kinase, which was so severe that it caused hemolytic anemia?
Yeah, that's I guess a couple of things. Yeah, that was the Vipracacid paper with Doug Hicks. Yeah, and so I guess 2 things is that indeed, you're exactly right. That relied on compound heterozygosity. So in other words, a mono, just a heterozygote doesn't exhibit a defect in pyruvate kinase. So I think that sort of is why maybe there's not so much stuff. It has been seen in some other papers, but I think one has to then be fortunate that you have both parents that can give you a compound heterozygosity in the patient, and that was actually looked at for pyruvate kinase. So I think the odds of that happening in some populations, you'd think it'd be more, it should come up more, but one could argue that maybe it is surprising that it hasn't been observed more than it has, given, yes, one can argue it's rare, but on the other hand, it does show up because you do have populations where you have loss of one allele of EKLF. So you would think their kids might have some of this, but no, but sorry, I can't answer more precisely there.
Because in red cell enzymes, it's very unique. Because that's the only time you have a pyruvate kinase deficiency or enzyme deficiency, which is caused by something totally distant from the gene which encodes that. Thank you, Jim.
Sure. Oh, yeah. So in terms of where EKLF is on this cluster, so again, just to go back, I just sort of wanted to leave this by saying F480 is expressed, although widely different, but KLF fell into just the clusters 4, 5, and 7. When you add the number of total, it's about 26%, which is not too far from what we'd seen before with the GFP markers, which was, you may recall, a little over 30%. But now, again, trying to say what is unique about these these 3 clusters where KLF1 is expressed, and maybe we can use that to pull these out specifically. So in this context, we looked for cell surface markers that we could use that were expressed in clusters 4, 5, and 7 that maybe could be useful for our purposes here. And we came up with a number of things, adducin 2, hemogenin, spectrin B, and this other gene called urexophyllin and PC esterase. These are highly enriched in those 3 populations. And indeed, when you look at the developmental profiles, it was totally cool. Just like EKLF, we have transient expression during early development. So this is, again, a dynamic population that is only present in this particular subset of macrophage cells. So, so using these, we went ahead and checked out the double positivity of the adducin 2 and the spectrin beta, because these were easily available cell surface antibodies. We get roughly similar percentages to what I showed you before. And when you look at erythroblastic islands, you see that these are enriched within the erythroblastic island, both spectrin and the adducin. And indeed, when you do image stream, you find that these single cells, which are GFP positive expressing EKLF, are F480 positive, are also positive. In this case, I'm showing you the adducin 2 data. Looking at this, then we can conclude that we believe we've been able to select markers that really pull out this particular subset in the fetal liver macrophage that maybe plays an important role in diagnosing the level of erythroblastic islands. I think I'll skip this. So when you look at these subpopulations from the single cell data, you can grossly group these into three large groups. And this is interesting because functionally they seem to fall into different Go terms. Like population 2 falls into this T cell activation and cytotoxicity. So this seems to be more of a macrophage function that's related to non really related to erythroid aspects. But now on the other hand, group 1 is involved in cell adhesion, has an enrichment for cell adhesion and iron. And group 3, which is where KLF1 and EPO are expressed, this is erythroid and myeloid, heme synthesis, and iron homeostasis. So when you look at these populations more carefully, again, on the y-axis, we have the identity based on these single genes that really are most diagnostic of each of these clusters. And we can find that there are 3 different patterns of expression. There's one pattern which is basically all of these features on the bottom are expressed all the way across. We have a second population where you have an enrichment of these genes. And finally, the third population, which is enriched, which is in the hemogen and HPG. So it looks like I have another question. Oh, okay.
It's actually a comment from Emery Bresnik.
Okay. Okay, great. So he's bringing up the point about the.
Jim, I didn't want to disrupt the flow, Jim. I just thought that Joe might not be aware of this finding that Pickler is a direct GATA1 target gene in normal heme environment. It suppresses the magnitude of the response. It's a direct transcriptional response. And nearly all of the genes that we study that are GATA1 targets often have KLF1 sitting at the genes. They're co-regulated. All the types of genes that you outlined at the beginning, the red cell antigens, the heme biosynthetic enzymes, et cetera, are all direct GATA1 target genes, and KLF1 collaborates with GATA1. But I wasn't sure if Joe, is he there? Yeah.
Yeah.
Was aware of this finding about Pickler and GATA1. So you can envision that any perturbation of the GATA system would certainly impact Pickler, and any perturbation of the KLF system would probably impact Pickler by disrupting this collaborative transcriptional mechanism. That was a lot more than I was intending to say, Jim, but
no, that's okay. No, that's interesting.
I know, I like the explanation too, Emery. Thank you.
So, so when you look at these gene patterns, you can see that they fall into three different types within here, and so KLF1 is over here on this pattern where you've got enrichment in cluster 3, but you can see these other genes are also enriched. And so what this one may hypothesize that you could even subselect based on the cell surface marker. This has a high level of VCAM, which is not really as obvious here, VCAM here. So one could select this subset of F480 macrophage cells that are VCAM positive, and these, when you look at the transcription factors, are enriched for MAF and NR1H3, whereas the genes, for example, that I told you, adducin-2, your spectrum beta, could be used to select for genes that are enriched in expression of EKLF and other downstream targets. So again, the overall idea would be to try to address whether one can then subdivide these heterogeneous populations and try to go after function. And EPO-R could be, as you can see here, EPO-R is definitely enriched in this pattern. So this goes back to what I mentioned earlier, looking at the single cell sequence data now, and looking where CSF1 receptor is, you'll note that it's actually low in populations 4 through 7, whereas EKLF was highest in populations 4, 5, and 7. So I think this idea of the Cree idea, I think I want to go back to doing that. That is knock out EKLF in the macrophage specifically and see the effects on the erythroid cell. But I think I want to do this now using a different Cree, if I can find one, that falls into this category where it's really enriched in these subpopulations. Because the CSF1 receptor Cree, and that maybe could explain why the effect was really sort of not as intense as I expected, as I had hoped. And so, so that's sort of one of the things I do want to end with here. So I think what I'd like to do, I think I should probably wind up here, which is that the model that we're focusing in a summary, what I'm telling you is that some of the properties are that the fetal liver macrophage is a unique cell type, is a transient EKLF positive population that arises during development at around E12.5, and that it regulates signature genes within the macrophage, such that one can come up with a model where you've got this intrinsic erythroid and extrinsic macrophage effects on the erythroblastic island, where EKLF regulates these signature genes, some of which enable an interaction with genes that are downstream of EKLF in the erythroid cell, for example, ICAM4. That interacts with alpha V integrin in the macrophage. Alpha 4 beta 1 interacts with, I'm sorry, and there's an interaction here, for example, with VCAM1, which I didn't list here because I'm a little unclear how that's involved here, might be involved here. And when you have disruption of EKLF, you have compromised cell interactions, as I've indicated, but also you have an inefficient removal of the nucleus. Do you remember that was something which was quite dramatic? And as a result, one maybe would be able to then try to parse this heterogeneity by selecting for some of these markers that I've indicated to you. This is not a view today since it's snowing here, but this is a view outside my window overlooking Central Park during the sunset during winter. And I use this to thank people in my lab. Kaustav Mukherjee was really went for the single cell analysis and all the really detailed macrophage analyses. Shui Li is the person who takes care of all the mouse studies. And Tanas, Sanjay, and Taslim are other people in the lab whose work I didn't get a chance to discuss. Jeffrey Glasberg is a collaborator on some other studies. This relied on some early studies for people in the lab from Deepa Manwani, Marianne Galdas, and Nithya when she was in the lab. And finally, our work is supported by NIDDK and HLBI and the Doris Duke Charitable Fund Foundation. Thank you very much.
Great. Thank you so much for your talk. Let's see. I'm seeing questions populated in the chat. Mark, do you want to go ahead and ask yours? Mark Perfetto?
Sure. Can you hear me?
Yeah.
Yeah.
Yeah. Have you guys tried adding an artificial nuclear localization signal to KLF1 to see if you can tease out the nuclear and cytoplasmic effects?
Yeah. Okay, KLF1 already has a very strong nuclear localization signal. That's why this has been complicated. So for example, when you transfect KLF, 100% of it goes into the nucleus. We've never been able to reproduce the nuclear cytoplasmic ratios by exogenous EKLF. So that's made it a bit more complicated. We've even done heterokaryon experiments, and we find that within the time frame of the experiment, all the EKLF from one nucleus goes into the other nucleus. So even with that, we can't time that correctly. But that would be nice if we had a way to analyze that. So I'm still unclear what it's doing there.
Hey Diane, I have a follow-up question to that one.
Do it.
So do you know if the KLF1 has some sort of a cell penetrating property where, you know, like FGF can go across the membrane and get into the cytoplasma or the extracellular space or cytoplasm of the other adjacent cells? Does KLF1 say in the cytoplasma can go through the cell membrane of erythrocyte and then get into the F480 macrophages where it starts activating all the genes?
Oh, that's an interesting. No, I can't say that we've done anything to directly address that. I mean, but in terms of getting into the F480, we know that the EKLF RNA and protein is already and is expressed in those cells. So I think in terms of, I know your point though is that maybe it could be coming from somewhere else, but we do see EKLF RNA in the isolated macrophage cells.
Okay.
And so I think one of the questions I see here is about phagocytosed red cell nuclei. That's definitely an issue in the field, and there's been some papers related to that. That's why I tried to emphasize that we really are careful. I think the use of this small peptide, which disrupts interactions, really has been helpful for that, because when we look at those cells, it's like 95% of them all have a single nucleus and they're macrophage. So it's not 100%. You could say, well, that 5%, but the thing is the extent to which we're trying to look at that, and also I think the image stream analysis was also very critical to say, okay, looking at individual cells, do we see any punctate, either red cell and macrophage or the other way around? And we don't see that. So I think I think we've done as best as we can to try to alleviate that concern. So but that's definitely a concern.
So F480 macrophages coming from the mainstream macrophages, or is it developed as a separate lineage?
My understanding is that it's not a separate lineage. I mean, from the original, from say the monocyte macrophage lineage, but I think it's a subset because when we look at the total, well, that doesn't really address your question. I was going to say, when we look at the total fetal liver, 9% of the fetal liver is F480 positive cells, but the only other thing I can address here maybe is that even many years ago, people found that MAC1, for example, was not a good marker for this erythroblastic island. So the MAC1 positive cells must be a different downstream lineage from the original monocyte macrophage. I think this heterogeneity issue is what really complicates the analysis.
Okay. But the EKL1 expression, well, so if I express the EKL1 in adult macrophage, can I induce the F440 from the adult macrophage?
Okay, that I don't know. I mean, right. So, for example, the role of EKLF in macrophage is way behind the role that we've looked at in the erythroid cell. So say, for example, upstream, would that be sufficient to give an F480 type genotype? That I can't say. That's an interesting thought. Yeah.
Okay,
We have
thanks.
2 other questions in the chat that we want to take, and then we'll wind up. Vanessa, do you want to go ahead and ask your second question there?
Oh, sure. Thanks. Dr. Bieker, this was an amazing talk, and I enjoyed listening so much. Thank you so much for this. Early in the talk, you had mentioned sort of a little bit of intrigue about the potential role for KLF1 in the bifurcation of MEP going to E and MK lineages. And I was just wondering if there was any clinical data from like patients who have mutations in KLF1 who are presenting with an erythropoietic phenotype, if they also demonstrate any kind of abnormalities in their platelet parameters to sort of further emphasize that potential role for KLF1 in the MEP.
Yeah, that's interesting. So I guess 2 things. One is, unfortunately, it's a mouse data, but there is a mouse model of anemia called the NAN mouse, the neonatal anemia mouse, and in those cells, it expresses in one allele. It's a heterozygous expression of a mutant KLF1 that alters its binding specificity. But so the red cells are screwed up. The mice are anemic. They've got splenomegaly, and interestingly, they've got higher than normal platelets. So that's interesting. But now switching to humans, say for example, like the CDA, congenital erythropoietic anemia patients, which have a mutation in KLF1, allele of KLF1. Unfortunately, I'm not aware that people have looked at platelet levels in those patients. The other angle could be like what we discussed before, where we have these compound heterozygous human patients. What's the level of their megakaryocyte or platelet levels? And unfortunately, I'm not aware that that's really been looked at directly. But yeah, that could be very interesting.
And then a really quick follow-up to that too, then. So if that pans out to be true, then I think it'd be really interesting to then have to tease out that cell autonomous effect of KLF1 in the MEP versus the effect of those mutant KLF1s on that systemic iron regulation, the systemic iron levels, which in and of themselves inherently influences MEP lineage output as well, right? Like,
yeah. So that would be the megakaryocyte erythroid progenitor. Yeah. Right. The closest. Yeah, but we didn't look at. It'd be good to follow up on experiments we did back then. One of which was but the question there was, does KLF1 repress megakaryopoiesis? So what we did is we misexpressed. We put KLF1 under the control of the PF4, I believe, promoter, and we made transgenic mice. So these mice were misexpressing EKLF. We took their bone marrow and we looked at megakaryocyte colony forming potential. And it was way down compared to transgenic mice that did not express KLF1 in the in the megakaryocyte. So that suggested to us that indeed it plays a repressive role in megakaryopoiesis. But the neat thing, which I didn't, at that point in time, we had a simulation. We had found that EKLF simulated at a single site. We had some hints that it's involved in repression. And we were asking ourselves, well, where could be their repression? Well, what about in megakaryopoiesis? So we did the same experiment with the transgenic mice, except now we misexpressed this single amino acid mutant, and it had no effect on megakaryopoiesis in the bone marrow compared to the wild type. So this is something we would love to follow up with. It's just been difficult to try to tease that apart, but that's sort of the overall. That's why it'd be neat, I think, with the CDA patients to try to look at. Anyway, I think I'm going on and on.
Yeah. No, it's fascinating. I appreciate you taking the time to talk about it.
There's one more question. Dean, do you want to ask your question?
Oh, sure. Just really quick. It may be something that's known and it's just outside my area, but I'm wondering in the adult, given that most of the iron is recycled from the spleen, do these erythroblastic island macrophages that you said provide a lot of iron for the developing red cells, how is that iron getting from the spleen to those particular macrophages? Is that known, or is it the very same macrophages? Do we know if they arise directly in the bone marrow, or are they coming from the periphery back from the spleen, for example?
Definitely the spleen also has erythroblastic islands. So there is, well, I should say in the mouse, since the spleen is an erythropoietic organ in the mouse, and I think it's controversial about in the humans whether you really do have extramedullary, but bone marrow also has the erythroblastic islands. But the thing is, where do those then get? Unfortunately, I don't know the answer. I mean, I would suspect that it does get recycled because I believe the macrophage also have the right receptors to bring in iron from. Probably some people in the audience would be able to better address that. But I do believe that they would be involved in the recycling process, not just downstream, like recycling iron. I mean, recycling the erythroid cell, but also upstream, getting iron from other sources, like maybe transferrin or other sources. But so I sort of having a gnawing there.
That's okay. I'm going to stop us now because we're just a little bit over. It's a wonderful talk. Thank you so very much. There are just two comments I was going to make. The first one is, I think, I don't know if Anna is still on or not, but she might be able to advise you on macrophage-specific Cree drivers that you might want to take a look at.
Oh, cool.
So Anna Bowden, and you could reach out to her. The second one is, there's a core that's being developed at Indiana University's medical school that's a Centers of Excellence core that's actually working with this technique called Codex, where they're working on trying to get erythroblastic islands in the bone marrow at least imaged, not live, that it's fixed, but where you can like pick up 8 or 10 or 20 different markers. It's a really cool technology. And for those who are interested in that kind of thing, I highly recommend you reach out to Ken Dunn at Indiana University. There's a video on the CCH.io website that you can learn about it, but he's really a great speaker, and you can reach out to him and he could help advise on the possibility of imaging those islands, to get you some more information, about proteins that are expressed on each of the different ones. And if KLF is in some cells, maybe actually moving out or something like that.
Yeah.
Anyway, super fun. Great talk. Thanks everybody for dropping in. This has been recorded and we'll make it available on the CIHD website, and hope to see you next month for our next speaker, who will be Kyle Hewitt from the University of Nebraska, who's going to speak to us on cis regulatory clues, reveal new mechanisms promoting cellular regeneration in anemia. So hope you can drop in.
Thank you very much. Thank
All
you.
right, and
everybody.
thank you for your
Enjoyed it.
presentation. Really appreciate it. Super
Thank
fun.
you.
Thanks,
you.
Great
Jim.
talk, Jim.
Thanks, Jim.
Everybody take
Oh,
care.
yeah.
Thank you.
Bye-bye.
Bye.
Bye-bye.